All right. Hi, everyone. For this next video on metastasis, we're going to talk about how we can transport a cancer cell safely within the bloodstream and then arrest in a capillary. In the previous video, we talked about intravasation, so our cancer cell getting into the bloodstream. And now we're talking about what actually happens inside the bloodstream when we've made it in. So if I draw my bloodstream right here, I'm going to have my cancer cell moving along. Maybe I'll move this down so I can write out what we're talking about here, which is transport and arrest. All right. So I have my cancer cell right here. And if we just leave it in the bloodstream without anything covering it, it has a very high likelihood of being recognized by immune cells. There's a much higher density of immune cells in our bloodstream. So let me just draw in an immune cell there. So we're going to need to actually cover our cancer cell with, in this case, platelets in order to be able to protect it from the immune system. So I'm just going to write my platelets as P's right here that are going to cover the entire cancer cell. And these are going to serve two purposes. The first is going to be evading immune detection, so what we just talked about, where now when this immune cell passes by this cancer cell, all it's going to see is a collection of platelets. It's not going to be able to tell that there's a cancer cell underneath that is kind of needing to be attacked by the immune system. Instead, it just sees platelets, which are normal to have in our bloodstream. So this is going to allow us to evade immune detection. The second thing or way that this is going to benefit us is if we have this cancer cell and it's moving along and it bumps into a wall. If it's covered in platelets, it's going to be protected from that mechanical stress. So that's kind of the second benefit, is that we're protecting it and cushioning it from mechanical stress. So you can kind of think of the platelets almost operating like bubble wrap. They keep the cancer cell safe from bumping into any of the walls or any mechanical stress that it can endure through kind of moving and transport in the bloodstream. And very few of our cancer cells that enter the bloodstream are going to survive, but the ones that do are going to then have to kind of arrest in a capillary and start to exit the bloodstream. And there's two different ways that this can happen. So the first way that we can exit a capillary is called receptor mediated. And in this situation, specific receptors on the cancer cell, so I'll put a receptor here, are going to interact with specific ligands that are present on the walls of the capillaries. So in this case, if this receptor bumped into this ligand, they would bind, and then our cancer cell would kind of be pulled to a halt. So that's receptor mediated, where a receptor on the cancer cell binds to a ligand in the capillary. The second way is that we can just have a mechanical arrest. So picture further down this blood vessel, maybe it gets a lot thinner. And as our cancer cell moves, let me draw my cancer cell again. There we go. This is my cancer cell. As it moves towards the end of this blood vessel, it's just going to get stuck here, right? And it's not going to be able to continue down that capillary. That would be the example of mechanical, where it just gets physically lodged inside the blood vessel and will start to exit the blood vessel. So those are our two different ways of exiting the blood and kind of the idea of how our cancer cells are able to survive in the bloodstream. In the next video, we'll talk about extravasation. So how do our cancer cells exit the bloodstream? What does that look like? And yeah.